Hi guys, this is a review video on Lab 9 for Histology, the microanatomy of the respiratory system. It's pretty long, but we're going to try our best. Basically, we are looking at the upper and lower respiratory systems, specifically the nasal cavity. We're going to look at the nasopharynx, at the back of the nasal cavity, the upper part of the pharynx, the larynx, trachea, bronchi, bronchioles, and this is all the upper conducting part of the respiratory system designed to get air into the respiratory part, which is going to be our respiratory bronchioles, the alveoli, and the alveolar ducts. So, let's get to it, starting with our nasal cavity. So this is the histo slide we'll be looking at. Importantly, it's kind of flipped upside down. This top side right here is towards the top of your head, and this part is the bottom of your feet. So what we're looking at is this section of the nasal cavity, specifically a cross-section that's looking here. So we have the nasal cavity is two paired chambers, right? You have this chamber, and it's separated by a bony and cartilaginous septum, which is this guy right here. Each of these two chambers has three regions. The first is the nostril, aka the vestibule region, which just means vestibule means opening, right? Then we have the respiratory region, which has our sinuses in it. These indentations or grooves we can see in the bone. And those are named based on, what bone they're embedded in. So you have maxillary sinus, there's a superior sinus up here, and we'll go over all of those. Then we have, if you look a tiny bit more at the this view of the nasal cavity, you can see in the center we have the nasal septum. We have these outcropping of bones that come in. There's one here, one here. These are called conchas. So this is right here, the superior concha, because our histo slide's flipped. And then at the bottom we have the inferior concha. And you can kind of see that. I think it might be this, not pointed out in the lab overall, so we don't have to worry about that too much. And then finally, we mentioned there's three regions to each chamber. We said vestibule, nostril, respiratory portion, which has our sinuses, and respiratory epithelium. And then we have our olfactory portion, which is the very top here, which is going to have our olfactory nerves, which will go through a plate right here into the olfactory bulb, which is connected to the brain. All right, so again, this slide right here is from this cross-section of the nasal cavity. All right, so starting with the nostril right here, we don't have this shown in our histology slide, so it's just important to know this is going to be lined with stratified squamous epithelium. It's going to be slightly keratinized, which means the top layers of the epithelium are going to not have nuclei. They'll be dead cells. There are going to be some sebaceous and sweat glands, some hair follicles. Just imagine your own nostrils, and maybe some of you have a lot of hair in that, that region. Anyways, the underlying connective tissue, so we have the epithelium, and then the underlying connective tissue, the lamina propria, blends with the perichondrium of the nasal cartilage. All right, so next place we're going to go is the respiratory region, which is shown in our histology slide. So basically, the respiratory region, the outermost part, aka the part that's above the bone and cartilage of the septum that you feel when you palpate, the ridge of your nose, it is going to be made up of, so the part that is on the inner surface of the cavity is going to be made up of mucosa, and that's going to be the case for really all of the different histology slides we talk about today. You're going to have mucosa, which is going to be made up of a respiratory epithelium, and then a lamina propria, which is loose connective tissue below it. All right, so respiratory epithelium in general, and again, respiratory epithelium is going to be here in the respiratory region. The olfactory region of the nasal cavity is not going to have respiratory epithelium. It's going to have olfactory epithelium, which is intended to do smell, right? Okay, so here, the respiratory epithelium is one part of our mucosa, the first part, the outer part. And so you can see that here, we have pseudostratified columnar cells that are ciliated. So our ciliated cells, they're going to beat synchronously in the posterior direction. They're wanting to expel material. We have our goblet cells, which you can see one here. Here's another goblet cell. They're lightly stained. They're going to produce mucus. Our basal cells are going to be down at the bottom of our basement membrane right here. So here's another one right there, the basal cell. And they can divide into other types of cells. Small granule cells are going to be there as well within this epithelium that makes the first part of our mucosa. But these really can't be picked out unless we stain them differently, so we don't have to worry about that on an exam. Same with the brush cells, which are cells that have microvilli on the top instead of cilia. And they are used for general sensation, but we won't be able to pick them out here. Again, this is the respiratory region of the nasal cavity. We're in this section, all right? So it is the respiratory part, it's going to have respiratory epithelium. So let's just take a closer look. As we said, for any of the things we talk about today, our outer layers are going to be made up of our mucosa, and then below the mucosa, you're going to have, in certain regions, a muscularis mucosa, and then a submucosa, and then bone. But overall, as we find mucosa right here, it's going to be lamina propria plus the respiratory epithelium. So this is our mucosa. And then later on, we're going to see we'll have smooth muscle submucosa underneath it. The lamina propria, loose connective tissue. We can see we have epithelium here. Lamina propria is deep to that. And then we have the periosteum, which is the edge of the bone. The lamina propria will contain mucus and serous glands. We know serous glands are for like watery secretions. The idea here is that those are going to leak to the surface and kind of clean the surface overall. Then we have some capillaries, very large veins, and those are intended. So here's a vein. Those are intended, the veins, the capillaries are intended to warm the air that is going through the nasal cavity before it gets to the lungs. All right, then we have of lamina propria, you can see, so if this is lamina propria right here, it starts to blend with the connective tissue of the periosteum, which surrounds bone, the layer that is on the outside bone. All right, so then we said that in this case, we have serous glands and mucus glands. We can point some of those out. Serous glands are going to be very darkly staining, and the cells are going to have some cytoplasm, but not a ton, right? So serous gland right here. Mucus gland is going to be light staining. It has relatively more cytoplasm, which you can see in those cells. It's important to know that we have our mucus gland, and then in our epithelium, you can see one here, we have a goblet cell. A goblet cell is a single cell that's producing mucus. A mucus gland is multiple cells together. Here's another one that are producing mucus, right? But the idea is that there's at least 20 different types of mucins, and so we have different types of mucus. Mucus is a glycoprotein, aka a mucin, with water, and so the mucus glands are going to produce a different mucus than the goblet cells, right? Another thing to point out here, we have these large vacuoles right there. These are lymphatic vessels. Okay. All right. So to recap, we have epithelium, lamina propria, and that together makes our mucosa. And then deep to that, we have connective tissue, the loose connective tissue of lamina propria starts to blend with the periosteum, and then you have the bone deep to that, which makes up the nasal septum. All right, so that brings us to our olfactory region. In the olfactory region, you can see that, right, this is the top of the nasal cavity. We have olfactory filaments, right, nerves that are going to be present in this region that are going to go up to, through the cribriform plate, this plate of bone, to the olfactory bulb. All right, it's important to know that these filaments are not myelinated. And you're like, these are nerves, why are they not myelinated? And the theory that was given to us in class was that they're not myelinated because of how close they are to the olfactory bulb, so they don't need to be myelinated for speed. They're relatively short nerves. All right, then we're going to be looking at our mucosa, which again is going to be epithelium plus lamina propria. All right, so the olfactory region, it's going to look a little bit different than the respiratory region. Respiratory region we said is respiratory epithelium. Olfactory region is olfactory epithelium. And so this is going to be exceptionally tall pseudostratified columnar cells, and there's going to be no goblet cells. So no goblet cells in the olfactory region. It's not going to be producing a lot of mucus, but you do have in this region a lot of other different cells. The exceptionally tall cells that we see are supporting cells that are going to support the olfactory cells. These supporting cells are called sustenticular cells, and they provide metabolic and physical support for the sensory cells. They are the most superficial nuclei in the epithelium. So all of these nuclei are the sustenticular cells, right? You can see their nuclei are here, but they go all the way down to the basement membrane, right? The basement membrane is right here, and then we have our basal cells down there, which are mitotically active, and then the olfactory sensory cells are bipolar neurons, which look like this. We can see them here, and they are going to send their dendrites through these olfactory filaments to the olfactory bulb. All right, and so once we're at the epithelium now, but we're going to look at the lamina propria, and we're going to see we have these nerve bundles. All right, so brush cells, again, not easily visualized. They have microvilli, so you can't really see them, but we see our basal cells here, which look a little bit like cones, and they rest on the basal membrane. All right, so then deep to the epithelium, right, we have the lamina propria that starts, and together those make up our mucosa again. Right, again, we're in the olfactory region. We're in this region that's going to send the nerves up to the olfactory bulb. So in our histology slide, if you go way back, so just to go back to the overview, right, this region here is our olfactory region. This region is going to be our respiratory region. So this is respiratory, this is olfactory. All right, and olfactory in particular is readily determined by the fact that its lamina propria is thick. All right, so let's take a look at that, because we looked at, oops, we went too far. All right, so the lamina propria is much thicker than the respiratory portion. It's going to have these Bowman glands that produce a watery secretion to moisten and cleanse the surface. We saw that here, right? So here's the lamina propria. You have the bottom of the Bowman's gland, and it's going to excrete onto the surface. It's going to be watery secretion to moisten and cleanse the surface. They also excrete lysozyme and secretory IgAs, aka antibodies, to help kill any, as an immune defense. We have those olfactory nerves present in this region. So first, here we can see Bowman's, all of these little tubules down here. Bowman's is going to, right, it's a branched tubulo-alveolar gland. So you can see that it's got these little alveoles, right, not to be confused with the alveoli in the lungs. This just means like, sort of bulbous. And then we see that it's in fact branched, and then it leads to the surface, right? But here we can see basically parts of Bowman's throughout the lamina propria. We have these huge veins here that kind of washed out. And then here's the edge of a nerve, which again is from we have the olfactory cells. Their nuclei are right here, and their axons are going to come back here and then travel through the cribriform plate into the olfactory bulb. All right, so lamina propria, deep to the epithelium. We saw our veins. We have Bowman's, these squiggly lines right here. Then we have our nerves, nerve bundles, and we have a lot of veins in this region as well to continue to warm the blood that's passing through this area. Also important to note the nerve bundles, a lot of these little nuclei we see are Schwann cells. If you remember, Schwann cells can myelinate one cell at a time, but they can also do organization, right? So we have a lot of Schwann cells that are organizing the axons that are passing through this region from the olfactory nerves that are in the center of the lamina propria and extending their dendrites and axons to the top. All right, so then that brings us to the pharynx. So we have the back of the nasal cavity with the nasopharynx, and then extends downwards to the top of the larynx and the esophagus. So the nasopharynx we're focused on. We're not going to be worried about the oropharynx or the hypopharynx. We'll get to those another time. But specifically, we didn't have any slides on the nasopharynx. Its epithelium is pseudostratified columnar, ciliated. So basically, it's respiratory epithelium. If you have abrasion, it might be stratified squamous. But it's going to have the cilia propel material towards the oropharynx in this case. It's lamina propria, mostly mucus glands. It's going to have a lot of lymphatic tissue, including the adenoids. All right, there will be some muscle in this region. Those are the pharyngeal constrictors. Again, I don't think this is something we really need to know for our exam, but it's good to know, at least in the sense that if you are asked about what would the epithelium of the pharynx or the nasopharynx be, you're just like, oh, well, it's respiratory. By default, I'm going to guess respiratory. All right, there's really going to only be two exceptions to respiratory. One is the olfactory region, right? And that's got the sustenticular cells instead of which are long cells, right? Sustenticular, on cylindrical, and they have microvilli at the top instead of cilia. All right, and the other exception will be the true vocal cords that we'll see in the larynx. All right, so in the larynx is the voice box, and we're now here, so we're below the pharynx. Larynx is really focused on vocalization, aka the voice box, and it's going to conduct air down to the trachea. So we're right here, we're going to go conduct air down to the trachea. Really, we're focused on the vocal folds, the true vocal folds, which are located here, and then we're also focused on the false vocal folds, which are up here, and they're big and fatty. And the true vocal folds, you can see we have vocal muscle, there's a skeletal muscle below it, and then we're going to be focused on the walls of the larynx, which we'll see as we get further down. But we're looking at a vertical cross section for histology. One of the ideas is that during swallowing movements, the cartilage are going to close the entrance to the larynx. So food can't enter. So we have this little piece of epiglottis right here, and that's going to move to close the entrance to the larynx so that the trachea is safe and no food can get in. All right, we'll be talking about the walls, and that's going to have supporting cartilage, interconnecting ligament that's fibroelastic between those two, intrinsic and extrinsic muscles. We're not going to look too much at that, but then of course we have our mucosal lining, which we know is epithelium plus lamina propria, and now we're going to start introducing the submucosa, which is going to be deep to the lamina propria and adjacent to the cartilage. All right, so then we have this histo slide from lab. This histo slide was like very confusing to me because you're like, what in the world are we looking at? This orientation is rotated 90 degrees, right? So here we have a piece of cartilage at the top right here. This cartilage is the epiglottis. So what we're looking at is a cross section that looks like this, and then that has been rotated to give us our slide that is like this, right? So this is the top end towards the head. This is the bottom end towards the trachea, towards your feet. And then really we have a bunch of different cavities. So this is our vesicle superior cavity space. It's going to be up here, below the epiglottis, which is here. Then we have our ventricle, aka the lateral cavity space. These are the ventricles right there in B. Then we have the rima glotitis. And so this is just, another cavity that gets closed, just like a ring. And so we call this entire region of the larynx the glotis, where the epiglottis is above it, but the glotis contains the vocal folds, vocal cords. All right. Then we have the infraglotic cavity below the glotis, and then deep to that we have inferior to that we have the trachea. We are focused today on the false vocal cords, which are right here, and then the true vocal cords, which are right here, right here. All right, we do have, so this is the epiglottis cartilage we said, and then we also have the thyroid gland in this region. So we have thyroid cartilage, and you can see part of the thyroid gland right here overall. All right, so then we have one last cartilage, the cricoid cartilage, which is right here. As we'll see as we get into the trachea cartilage, deep to this is going to be C-shaped. The cricoid cartilage is a complete ring, right? It's one complete ring. This is the cricoid cartilage. Do we need to know this? No. Then we're going to deal with the wall of the trachea overall. All right, so the wall of the larynx. Larynx is what we're focused on. Trachea is next. All right, so the layers of the wall of the larynx, we have supporting cartilage as the deepest part, the interconnecting ligaments between those. We have some muscle in this region. We're focused on the mucosa, and then deep past the cartilage is going to be the adventitia. And we'll see that, but our mucosa is our epithelium plus our lamina propria. Right, good rule of thumb for everything here is that we have respiratory epithelium, right? We said our exceptions to respiratory epithelium are olfactory part of the nasal cavity, and then we're going to see that here it's all respiratory epithelium except for the true vocal cords. All right, so here our mucosa, it's going to be pseudostratified ciliated columnar, aka respiratory. It's going to have goblet cells, except for in regions of abrasion. So if you have a lot of abrasion, then you can have it transform into non-keratinized stratified squamous epithelium. This is also, when this happens, this is called squamous metaplasia. Our ventricular false folds are going to be that same respiratory epithelium, but our true vocal cords, where you have a lot of abrasion, is going to be stratified squamous non-keratinized. So this is our only non-respiratory. But here you can see this is a weirdly stained goblet cell right there. Then you can see that the epithelium is in fact pseudostratified ciliated columnar. And then we have our basement membrane right here, our basal cells. Deep to that is going to be our lamina propria, which remember is loose connective tissue. And then past that, we're going to start to see our submucosa. So we'll get to that in just a second. All right, so looking at the mucosal lining, right, we have our respiratory is on the walls of the larynx, there's more on this, lamina propria, and then deep to that our submucosa is going to be collagen and adipose. You can see there's a decent amount of adipose here, but loose connective tissue, and then importantly, it's adjacent to the cartilage, right? So right next to the cartilage is going to be submucosa. It also contains serous and mucus glands. So this one appears to be a, this looks like serous to me, but this could be some mucus, so this could be like a mixed submucosal, but the in this region, our submucosa is going to have many mucus glands and few serous glands. All right, and then deep to all of this, so we have our cartilage. We said past the cartilage is our adventitia, and this is just going to be a whole lot of fat and loose connective tissue to anchor. To past that, we have muscle, right? So this is the lumen of our larynx. And then as an important note on our slide, all of these little red bubbles, this is the thyroid gland. All right, so larynx, the true vocal cords, so now we're located right here. This is the one exception, that's the non-respiratory section, other than our olfactory region of the nasal cavity. This is going to be simple squamous epithelium. Excuse me, this is a typo. This should be stratified squamous, as we can see here. This is in fact stratified. And deep to that, we have a very cellular lamina propria with loose connective tissue. And then our submucosa is going to have a fibrolactive connective tissue running through it, which we call the vocal ligament. And the idea is that this submucosa right here is going to anchor the mucosa to the vocalis muscle, which is skeletal muscle, deep to the submucosa. All right, but fibroelastic connective tissue in the submucosa region, how can you separate the two? Well, you can kind of trace here where these hypercellulated, or there's a lot of cells in lamina propria here, so where you go from high concentration of cells to more spread out, then you've gone from lamina propria to submucosa. All right, so again, mucosa, submucosa. And the submucosa here is going to have a vocal ligament, which is fibroelastic connective tissue. It's not well shown here. I think we need a different stain. All right, then we have our false vocal cords, aka vestibular, aka ventricular. Why would you call it that? Remember, this is the vestibule. This is the ventricular cavity, vestibular cavity, ventricular cavity. Important to note, it is in fact, again, respiratory epithelium. If you have friction, you could have stratified squamous, but how can we identify this? It's just an enormous amount of adipose tissue in the submucosa. Again, our lamina propria in this region has a lot of cells, and then we see here epithelium overall. All right, so that brings us, we are now done with the larynx. The bottom of the larynx is the trachea, and the trachea extends to our left and right bronchi, which go into the lungs. All right, so our trachea is a 10-centimeter conducting tube, aka just means air goes through it, and it maintains patency with cartilaginous rings, C-rings overall. Remember, we said that the cricoid ring in the larynx was the one ring that is actually a ring. Everyone else for cartilage is going to be a C shape, not a complete circle. All right, so wall structure for the trachea is going to be made up of our friend, the mucosa epithelium plus lamina propria, which is our loose connective tissue, which you can see right here. This is our friend respiratory epithelium, pseudostratified ciliated columnar. We have our, that's right there, then our loose connective lamina propria. We do have a submucosa in this case with our seromucous glands and loose connective tissue. We also love submucosa because you can find it easily because it's adjacent to the cartilaginous layer, and we can see that we have a nice cartilaginous layer here with our C rings that maintain patency, aka keep it open. Patency is remaining open. All right, then we have deep to that our adventitia, which is loose connective tissue and some fat. All right, but we're in this region now, and let's get to it. So submucosa, cartilaginous layer, adventitia. All right, so the trachea, we're looking at sort of a coronal or frontal cross section of this. Again, this is the same slide as the larynx, the vocal cords, right? It's just the bottom of the larynx starts to be trachea, right? So we're seeing a frontal cut, right, just like cutting through the corona of your eyes. And so looking at this, our mucosa, you can see is in fact respiratory. We have cilia. We have a very thin loose connective tissue region that has a dense number of cells. That's our lamina propria. But the trachea is special because it has an exceptionally thick basement membrane. All right, so you can see here we have our respiratory epithelium, but this guy right here is like the chunkiest basement membrane you'll ever see. And so if you see that, you're like, okay, I've gone from larynx to trachea. That is one beefy basement membrane. All right, so mucosa, epithelium, thin loose connective tissue for lamina propria, very prominent basement membrane. Our submucosa, we know of course, is adjacent to the cartilage. So you start there, and then you go to really, you often see the start of a gland, which you know is submucosa with the serous mucus glands, or you can try to go based off of the density of the cells in the lamina propria, but the submucosa is loose connective tissue. It's denser than lamina propria. It's going to have those mixed seromucus glands, which you can see here, and then it's going to be separated from mucosa by thin elastic tissue. It's not evident in this case. We need a different stain. We have our cartilage layer here, which you can see. The C rings, they're haline cartilage, which we know is type 2 collagen, and they maintain the openness. In between the adjacent rings, so here's one ring, here's another ring, they're separated by fibroelastic connective tissue, blending their chondrium. So in this region, you have fibroelastic connective tissue, and then here we have the thyroid gland right there, and then we have our adventitia up here, adventitia external to the hyaline cartilage, and connective tissue, blood vessels, nerves, adipose. All right, cross section of the trachea. So we're looking at a coronal view here. Cross section, though, we can see a nice C ring, right? It's not a complete ring. And at the opening of the C, you have the trachealis muscle, which is smooth muscle. We do have, of course, a respiratory epithelium. There's no exception to our rule. We have our lamina propria, loose connective tissue, submucosa with our glands, seromucous glands, and then past the cartilaginous ring, cartilaginous layer, we have our adventitia with our loose connective tissue, adipose, nerves, veins, etc. All right, so here's just a closer cross-section, mucosa. We see we have respiratory. We have an enormous number of goblet cells here. So lots of goblets. Okay. All right, very prominent basement membrane, of course, which is a little hard to see in this, but you can see it right there. Then we have deep to our lamina propria. We have, and adjacent to our cartilaginous ring, we have our submucosa layer. And again, good hint is you're going to have your seromucous glands, and then a little bit past that is going to be the start of your submucosa. It's loose connective tissue, but it's denser than the lamina propria. And again, there's that thin elastic layer separating mucosa from submucosa. We have our cartilaginous ring and our adventitia here. All right, so moving on. Just additional look at the individual cell types. So we said that we have an enormous number of goblet cells. You can see those right here. They're producing that mucus onto the surface. We have a lot of ciliated columnar cells, right? This is respiratory epithelium. So here's a nice ciliated columnar cell. We have the standard brush cells that have microvilli. You can see an example in this diagram here with the brush cell. And again, what are brush cells used for? They're general sensation cells, but we generally can't see them very well in H&E stain, so we shouldn't have to identify them. Then we, of course, have our lovely cone-shaped basal cells, which are mitotically active and can divide into other types of cells. All right, again, thick basement membrane, our friend, the trachea. All right, so past the trachea, we start to get to our bronchi, and we have a whole bronchial tree, so we'll start to get into that, but the place that you first start is you have, remember from dissection, you have right, the hilum and the root of the lung, and you actually have bronchi that's external to that, so you have the extrapulmonary branches, which are the left and right main primary bronchi, which you can see here and here. They're going to be exactly the same as the trachea in terms of histology, these extrapulmonary branches, and then once you get into the lung itself, you have the intrapulmonary bronchi, and then you have, remember, we have lobes of the lung. So the primary bronchi, once they get in, are going to start to separate into secondary bronchi, and each lobe of a lung, right, the right lung has 3 lobes, the left lung has 2 lobes, is going to be served by a secondary bronchi. Those are going to separate into tertiary bronchi, which supply the bronchopulmonary segments of the lung, right? There's 10 segments in the right lung, 8 to 10 in the left, but the histologically, what's going to happen is you're going to go from, basically the layers of the intrapulmonary bronchi are similar to the trachea, except for the epithelium is going to go from traditional pseudostratified ciliated columnar to, instead of pseudostratified, simple ciliated columnar, and it will continue to have the goblet cells, right? Basal cells and endocrine cells are also present. So just a tiny bit more about the respiratory tree before we move on. So we have our primary bronchus, which are outside of the lungs. Then you get to inside the lungs, you have our secondary bronchus, which we said is one for each lobe. Those break into the tertiary bronchus. Then you have past that, you're going to divide into lobar bronchioles. Lobar bronchioles are going to divide into, so we have two terminal bronchioles, and these are the last true bronchioles because they're going to have bronchial epithelium, right, which is we're going to simple columnar, this is pseudostratified, and then the last true bronchioles, aka the terminal bronchioles, are going to give rise to respiratory bronchiole, and respiratory bronchioles are not called terminal because they're like part bronchial epithelium and then part alveolar lining. All right, and they define the smallest functional unit of the lung, which we call the pulmonary acinus. And so, right, you have terminal bronchiole, the last true bronchioles, bronchial epithelium, then you get to the respiratory bronchiole, and the respiratory bronchiole defines the pulmonary acinus, and it's going to be, you can see already, there's alveoli sacs here, so you're going to have bronchial epithelium, but then you're going to also start to have alveolar epithelium present, right? And so that's why it's not called, that's why the terminal bronchiole is called terminal, because it's the last true bronchiole in the sense that just bronchial epithelium. All right, pulmonary acinus we'll talk about more, but it's defined by the respiratory bronchiole, which is right partially bronchial epithelium, part acinar epithelium. In there is going to be mostly cuboidal, but it's going to contain club cells. The lamina propria is going to have elastic fibers, and then if you lose elasticity in the alveolar sac region, you're going to have something called emphysema, which is characterized by large interalveolar spaces. This is bad. You want to have as much surface area as possible, so you want to maintain elasticity, and then there's going to be smooth muscle around the respiratory bronchioles. All right, so let's get into it. Starting with the secondary and tertiary bronchioles, we said that the bronchioles are going to look like trachea, except for they're going to have slightly different epithelium. You're going to go from pseudostratified to just simple columnar. So we can see that in our mucosa, pseudostratified columnar to simple columnar ciliated. The lamina propria is going to be just a very thin layer of loose connective tissue. You have this new muscularis mucosa, which is considered part of the mucosa. It's interlacing bundles of smooth muscle that separate the mucosa from the submucosa. The submucosa is collagenous connective tissue with mixed seromucous glands. We're familiar with this. We've seen it since the larynx. And then cartilaginous layer is going to be hyaline cartilage type 2. Importantly, the bronchus is not going to be nice C-rings, so our C-rings are gone. Instead, we have plates. Okay. Then finally, past the plates, we have our adventitia, loose connective tissue external to the cartilage that blends with the lung tissue. All right, so how do we get started? The first start is if you get a histoslide that's clearly lungs, right? This is clearly lungs. You can see there's like so many different spaces. If they're like, what is this guy and what is this guy? Well, the telltale sign that you're dealing with a bronchus, either secondary or tertiary, is going to be haline plates, right? You're like, how would I know? Like, what if this was a primary bronchus? Well, primary bronchus is extra pulmonary, meaning outside the lungs. And we said primary bronchus look exactly like trachea. Primary bronchus are going to have C-rings. There's no C-ring here. There's no C-ring here. But you do have these discontinuous cartilaginous plates, which tells you are secondary or tertiary bronchus. All right, so the cartilaginous plates is your big hint that you are secondary or tertiary bronchus. All right, so that's how we know this guy and this guy are either secondary or tertiary. This guy right here does not have cartilage, so this is probably bronchial. Or it's got to be bronchial. All right, so another cross-section here. None of these are the greatest pictures, so we're going to zoom in a little bit later, but here we can see ciliated simple columnar epithelium that's in our intrapulmonary bronchus instead of pseudostratified. Then we have deep to that are going to be our thin lamina propria, which we can see over here. So you're like, is this pseudostratified or not? Well, just try and trace out a couple. It gets tricky because, right, you have the basement membrane here, and you're like, what is going on? Is this, right, this is, you do have basal cells. So this is going to be our simple columnar. And you can see it's still ciliated. Deep to that, we have our lamina propria in this region. Then you have the smooth muscle layer, which is new in the final part of our mucosa. Deep to that, you have the submucosa, which we always know is adjacent to our cartilage, so that's our big hint, but it's loose connective tissue, and then it has our seromucous glands in it. This image here, sorry, is not super useful, it's just too zoomed out. And then past this, we have our adventitia, and here's a good example of our adventitia, which is past our cartilaginous layer. All right, so after we go through our bronchus, right, so after tertiary bronchus, you get to our first bronchiole, so our lobar bronchioles, and they're not going to have cartilage, right? So this guy right here is probably a lobar bronchiole. There's no cartilage around it. Thin walls, no cartilage, no submucosal glands, and it will have some smooth muscle around it, which you can see right here. This is going to be bronchial epithelium. So again, we've moved on from respiratory epithelium. We now have simple epithelium, right? Not pseudostratified. So simple columnar or simple cuboidal epithelium with ciliated cells throughout. At this point, we've reached far enough into the lungs. We've gone past our situation where having mucus is going to be useful. So we do not have any goblet cells. However, we do have club or Clara cells, which are these dome-shaped cells in our epithelium. They secrete one of the components of surfactant, but they don't secrete surfactant itself, which is a kind of annoying distinction. And they're important for chlorine ion transport, which is going to have water follow into the lumen, which aids in keeping the lumen surface moist. And they can act as reserve cells. They're capable of dividing to other cells and replacing damaged cells. And then they contain enzymes and enzyme systems that detoxify noxious substances. All right, so again, you look for this dome shape right here, but our bronchiole, these lobar bronchioles, are going to have our basement membrane. And then if you were to try and box these, they should be approximately cuboidal, or they should be simple columnar, and they're going to be simple, right, one layer. And then deep to that, we have a little bit of lamina propria that's below the basement membrane. So here's a lamina propria, loose connective tissue, very thin. It will have some elastic fibers, and then deep to that, we can see that we have the muscularis mucosa layer right here, very thin layer of smooth muscle. All right, important to note, there's no submucosa here, right? There's no cartilage, right, which goes with submucosa, so none of that. And then there will be some loose connective adventitia, though, that is past the muscularis mucosa layer that blends with the lung tissue. All right, so then after the lobar bronchioles, we go to the terminal bronchioles, which are another division. So we have terminal bronchioles, which will eventually give rise to our respiratory bronchioles. And then the respiratory bronchioles, remember, makes up our acinus, which we mispronounced as acinus. All right, so our terminal bronchioles, or last true bronchioles, basically the way that you identify these is by recognizing that it's going to branch and give off respiratory epithelium. So it is respiratory, or is bronchial epithelium, so it's not the traditional respiratory epithelium, it's ciliated cuboidal epithelium, which is known as bronchial epithelium. It's going to have those club cells, which again act as reserve cells. They do chlorine ion transport to get water to moisten the surface of the lumen. They do secrete one of the components of surfactant, and then they have enzymes that can detoxify not just substances, but importantly, again, we're focused on how to identify these, the fact that it gives rise to respiratory bronchioles. All right, so we have this image from class, and the trick here is to follow the basement membrane. So you can see here's the basement membrane. All right, and I'm doing it until it stops, right? And then there is some basement membrane here, it stops, and I think there might be some here, but there's clearly a pouch here, and then there appears to be some kind of pouch here. So the terminal bronchioles are the last true bronchioles, so that means that its entire epithelium is going to be bronchial. Here you have a case where you have a mix of bronchial epithelium, and then you have, these are alveolar epithelium. So this guy right here and this guy right here that have these outpouchings are going to be respiratory bronchioles, and this guy that has purely bronchial epithelium is going to be the terminal bronchiole. All right, so looking at the basement membrane and then seeing where it falls off is going to allow you to find branching, and then the part that has a mix of, this basement membrane that is a sign of the bronchial epithelium, the ciliated cuboidal epithelium with club cells, and a mix of that with the alveolar epithelium, that's a sign of respiratory bronchioles. The part that is only bronchial epithelium is going to be our terminal bronchiole. All right, so respiratory bronchioles are, this section right here. We've pretty much already said it, but the epithelium of a respiratory bronchiole is going to be made up of, right on this side we have bronchiolar epithelium, and then on this side we have alveolar epithelium, which is going to be squamous cells with type 1 pneumocytes, which do gas exchange, and then type 2 pneumocytes, which are going to do surface production, right? And so if we look here, we can see we have our terminal bronchiole. This is all bronchiolar epithelium. Then we get to our respiratory bronchiole. You can see that we have bronchial epithelium, and then we have acinar overall. And then we'll see that the acinar is made up of these type 1 pneumocytes, which are simple squamous, which are going to define the walls of our alveolar sacs, right? The challenge is they are very thin. We were told that the alveolar type 1 pneumocytes are super spread out, meaning you'll see like one nucleus, and then its cytoplasm extends over an enormous surface area, right? Which is what we want, because we want to have as much surface area in contact with gases to allow diffusion of that gas through the cell into the capillaries that permeate the lung, right? So it does make it a little bit challenging, but this brings us to alveolar ducts and alveolar sacs. Our respiratory bronchiole is going to be half and half bronchial. Alveolar ducts are going to be full alveolar epithelium, so you can see that here, right? We don't have any columnar, cuboidal, ciliated epithelium at all, so that tells you that you're an alveolar duct. And then you get to these outpouchings, which are going to be our alveolar sacs. And these alveolar sacs are lined by epithelium, alveolar epithelium, just like the alveolar ducts. They are squamous cells that are really our type 1 pneumocytes, which are the gas exchange cells. They're squamous with very thin spread out cytoplasm. And then our type 2 pneumocytes do surfactant production. And then we also have alveolar macrophages to eat up things that are not supposed to be there, right? Macrophages eat up bacteria. They also eat up, any tiny little bits of foreign material, or like tiny pieces of smoke, etc. All right, so here we have some examples. Right here is a big fat great alveolar cell. It's type 2. This is doing our surfactant production. So here's one, and here's another one. Then we have, and how can you tell that from the type 1? Well, type 1 is going to look like a flat pancake, right? It's squamous. So here's a flat pancake right here, and I think that's the only one, but they have exceptionally long cytoplasm, right? So that's why they're not super easy to identify, but they are squamous. So if it's like super flat and you're clearly in like lung tissue here with no ciliated columnar epithelium, you're like, okay, I'm clearly in an alveolar duct, alveolar sac region. Then you're like, I see a squamous cell, that's going to be my type 1 alveolar cell, which does gas exchange, all right? Then you look for, on the surface, we have our nice fat macrophages, which, have a whole lot of cytoplasm. They're large cells. And then also on the walls, we have our type 1s, which are going to have a big fat nucleus, and they do our surfactant production. All right, so moving on, in the alveolar wall, right? So we have on the outside type 1 pneumocytes, which have very spread out, but inside of this, you want the gas to go through the type 1 alveolar cell and get into the wall where they should meet some capillaries, right? And so this is an electron microscopy of an alveolar wall. It is terribly labeled, but important to know, here's a capillary right there. And here's like the nucleus of the endothelial cell that is making that capillary. We have, this is the alveolar lumen, this region right here. Then we have gas exchange barrier, so this looks like the nucleus of the type 1. And then this is cytoplasm of type 1 right here. And then you have right here, this is the fused basal membrane of epithelium and endothelium of the capillary. So you can have a fused type 1 and the endothelium of the capillary can fuse to make gas exchange easier. All right. Also in this region, in the cell walls, you have fibroblasts, and then you can also have our macrophages get into the cell walls as well. All right. And then important to note, we have these things called the pores of Kohn, and they allow for pressure equalization between alveoli and provide collateral air circulations in case of bronchial obstruction. All right. Do we have a good example of that on here? Like I don't see one. Maybe right here there's one, but we won't actually point that out in lab. All right. So now it's time for the lab review that comes to the lab guide. We go back to our trachea. We also think to ourselves, we love the trachea. Why? Because it's that thick, fat basal membrane, which is right here. Of course, we're so zoomed out we can't see it, but we want to identify layers of the wall of the trachea. Trachea is our respiratory epithelium, and it's got those C cartilage rings, right, to keep it open. So on the outside, we have our epithelium, which we can see right here. Deep to the epithelium, we have our lamina propria, loose connective tissue, relatively thin layer. Deep to that, we have our submucosa, which is loose connective tissue plus seromucous glands. And we know that's adjacent with the cartilage, so we have cartilage here. Then we have past that our adventitia, which is loose connective tissue, adipose, and veins, nerves, capillaries. All right, parts of the tracheal epithelium just sort of zoomed in. Again, the big fat basement membrane, it's a basal cell. Big fat basement membrane, here's a nice basal cell. We have in our trachea an enormous number of goblet cells. So important to know, trachea, enormous number of goblet cells, also big fat basement membrane. All right, we also have our typical ciliated cells. Then we have, so lamina propria, which is right here, loose connective tissue. Then we have past that the submucosa right here, which we know is adjacent to hyaline cartilage. It's loose connective tissue, but it's going to have more dense connective tissue. It's going to be denser than the lamina propria. That said, lamina propria often has a lot of cell nuclei. All right, adventitia we can't see here, so we'll move on. Electron microscopy of tracheal epithelium. So we have our ciliated columnar cells. You can see right there, tracheal epithelium is respiratory epithelium. Love it. We have an enormous number of goblet cells in this region. So here's a nice goblet cell producing its mucus onto the surface. Then we have our basal cells, which we said are roughly cone-shaped. They are mitotically active. We have our non-ciliated columnar cells, aka the brush cells for generalization, not shown here. They never seem to be shown. And then we have our small granular cells, and there is one located here. And unfortunately, it doesn't seem like we can see our granules super well, so hopefully that's not a test question, but we can see that it is along the basement membrane, which is a good hint, and it's not short and fat like the basal cell. So if you're like, okay, it's stuck to the basal membrane, it doesn't go all the way to the top, then you're like, it's probably a granule cell. Hopefully you can see some granules. If not, it's kind of mean. All right, so bronchial epithelium. So we have, again, this is bronchiolar, meaning not bronchus. We have our club cells, so, or this is a Claire cell, formerly called Claire, now called club cells. We have the round dome shape, which is our big hint, produces watery secretions through the chlorine ion transduction, or like water follows chlorine ions. So if you pump chlorine ions out, water's going to follow it onto the surface. They are reserve cells. They can divide into other things, and they produce a part of what makes up surfactant, although not surfactant itself. We're told that in lecture. Here we have a cross section of an alveolar wall. What's a good hint here? Should an alveolar wall have a capillary for gas exchange? It certainly should. And then we have the endothelial cell of the capillary, and we can see that blends its cytoplasm with the cytoplasm of the type 1, right? So blending here, type 1 alveolar cell type, type 1, oh my gosh, I'm blanking on the name. Type 1 alveolar cell. Here we have a type 2 alveolar cell, which we know does surfactant release, and then we have a fibroblast right here. I'm not sure exactly how you would tell it's a fibroblast. Fibroblasts are supposed to be spindle-shaped. Is it kind of spindle-shaped like a football? It's bigger in the center, narrower at the ends, maybe. All right, and then here's our final picture. This is just a type 2 alveolar cell doing surfactant production. So you can see that we have choline, amino acid, the rough endoplasmic reticulum, protein synthesis going to the Golgi, producing vesicles full of protein. Then you're fusing into some lipoprotein vesicles, and then that's going to be excreted onto the surface of the cell. And then we can see we have type 1 cells are also in part of the epithelium of the alveolar sac. All right, and that's it. That's the end. That was super long. I feel like I'm out of breath, but good job, guys, and we'll see you next time.